So by using a different combination, or a combination of different restriction enzymes, scientists can actually examine smaller pieces of DNA and then slowly piece them back together to view the whole. This is especially useful with bacterial DNA, which is actually circular. So that's what we're going to be looking at here. Okay, so they did 3 different digests with copies of the same piece of DNA. They digested it with HPA1 and then Hindi3 is actually how you say that. So 2 different restriction enzymes here. They did a digest with each one individually, and then they also did a digest with both of them together. Okay, and then ran those results on a gel. When they got that, they got different fragments obtained. So for the HPA1, they just got a 26 kilobase pair length segment or fragment. Okay. For Hindi3, they got 13, 6, 4, and 3. Okay, which if you'll notice add up to 26. And then when they digested them both, okay, they got they got a 7, 2 6s, a 4, and a 3. So, as this is a bacterial piece of DNA, it is circular, so we'll start with our circle here. Okay, so we know that there is 1 restriction site for HPA1. And we know that because it only produced 1 fragment. So it's basically splitting this circle in the middle somewhere, and then you just get 1 line. Okay. We also know that this entire circle has about 26 kilobase pairs. Okay, because of this here. Okay. And then we can also start to fill in the Hindi3. Okay. So we know that Hindi3 has 4 different restriction sites because it produced 4 fragments. So we have our 13 kilobase pairs, 6, 4, and 3. Okay? And that's just kind of arbitrary. You just kind of put them around. If you wanted to get more and more precise, you would actually have to do a lot more digest with this. But okay, so now we have all 4 of our Hindi3 restriction sites mapped onto this diagram. So now we need to figure out where the HPA1 restriction site is. Okay, so if we look, you want to look for the differences between these 2 results. Okay, so the 4 and the 3 are the same, so we know that it's not here. We do have a 6 kilobase pair in both of them, so it's not here. But then you have another 6 and a 7, which if you add those together equals your 13 kilobase pairs. So your HPA1 restriction site is actually in the middle of this 13 kilobase pair fragment here. Okay? And you've got your 6 kilobase pairs and your 7 underneath that. So now looking at it a little bit backwards. So this would be if a scientist did all of this work and then lost their actual results of the digest. Okay. So we know that we have 3 different digests performed. We have one that just has EcoR1, and then one that has just BAM-HI, and one that has both EcoR1 and BAM-HI. Okay? So, easy ones. Okay? You can see with both of your restriction enzymes, you have 8 kilobase pairs, 4, 2, and 6. Going around the circle this way. Okay? Now with your EcoR1, okay, you only have 1 restriction site for it, which means that you just add up all of these numbers, and you have 20 kilobase pairs total for this piece of DNA. And then with the BAM-HI, all you do is just knock out the EcoR1, okay, and add these 2 together, because now they're going to be a single piece. So you're going to have 2, 6, and now 12. Okay? And that is restriction mapping.